Good morning. I am Dr. Srinivas Bundaru. Today we'll be dealing with molecular biology, and let's come to the session 1, which is structure and organization of the genome. So you see, once the learning outcomes are, you'll remember what's the organization of the genome in both prokaryotes and eukaryotes, and remember the variation in complexity. You have different complexities in prokaryotes and eukaryotes. So what do you call a genome? So when anywhere where you are finding in the in terms of life sciences, where you are finding some parlance of ohm, it's a collection, right? So when I'm saying it's a collection of all the genes, you call it genome. And the same way, if you have all the proteins together, you call it proteome. So anyway, so genome is all the DNA in the cell. Here what we are talking about eukaryotes and prokaryotes, where when you are finding some RNAs, which is also a genome in some viruses. We are not talking about that right now. So all the DNA are all on the chromosomes, and which includes genes and intergenic sequences and repeats. Specifically, it's the DNA and it's in the organelle. Fine. So eukaryotes can have 2-3 different genomes. Not only one genome. It's like you have nuclear genome, which is constitutes almost 99% of that. Then you have mitochondrial genome, and also when you talk about plant genome, you have plastic genome. So it's not specified. Genome usually refers to the nuclear genome. It's not that. So when you are talking about the genome as a whole, it can include the nuclear genome, mitochondrial genome, and plastic genome, and so on. So if you happen to see the components of the human genome, in particular, when it was sequenced, and first draft of human genome project came in 2002. So what it says is it has around 3.2 billion base pairs of DNA, and only 3% of the code codes for the proteins, right? So those which is the coding for mRNAs, and 40 to 50% are repetitive, and they also have a retroposition. So what is the function of the remaining 50, or I should say more than 80%? So what's the function of that? It helps in regulation, and the discovery is still going on. So you see, the genome is thus the intercollection of whole genes, including RNAs, which is coding for mRNAs, not only which is coding for mRNA, which is coding sequences, also tRNAs, non-coding RNAs like tRNAs, and so on. Then it can have functional sequences like regulatory sequences, or initiation sites and operator sites, and so on. Then you have non-functional sequences. Today we are calling non-functional sequences. Okay, earlier it used to call as non-functional sequences, but you have different types of functions of that. For example, if you see between the introns, it helps in the splice site mechanisms, right? So which is helpful in needed for coding, regulation, and replication of the DNA. Just a comparative genome sizes of organism. Let's have a comparative look. So let us take a reference as Homo sapien. You see it's 3.2 billion bases, and the number of chromosomes is 46, and mouse is around 2.6 billion bases. Same way you see E. coli, it's only 4.6 million, and you have a certain chromosome numbers to that. So when we do some comparative genomic studies, let us see how we can compare only in the terms of genome. So human, when we take human, is around 40% similar to the mouse, 4% similar to Drosophila melanogaster in terms of the genomic sequences, and 1% similar to E. coli, and 99% similar to the chimpanzees. So you have a comparative genome sizes. However, having just a genome size doesn't make a whole story. It's all the transcriptome, which we'll be discussing later. So you see, you have prokaryote and eukaryotic genomes when you are finding. So it is prokaryotes, of course, as it's a single cell, and eukaryotes you have a single or multi-cell, like yeast you have single cell and multi-cell like humans are having. So in prokaryotes there is no defined nucleus, what you call a nucleoid, right? And what you are finding in eukaryotes is a nucleus. Okay, and there is a one piece of circular DNA in the bacteria or in the prokaryotes generally, and also you have additional genome like plasmids and so on, and all the genome is actually organized in chromosomes in eukaryotes. So you don't have much more of post-transcriptional modifications when you are seeing in prokaryotes. But yes, in eukaryotes you have all those post-translational modifications, including 3' and pi dash modifications in the RNA and splicing and so on. So you see just a prokaryotic genome, it's a 4.6 4 into 10 to the power 6 bases, and more than 90% of the DNA encode for the protein. That is very simplified. It's not much complex when you compare it with the eukaryotes, and there is no histones present, which helps in packaging. You of course don't require it, because what you're finding in the eukaryotes, the genome is too big, so you need a proper packaging system there. So eukaryote, you see you have a nucleus. If it's a very well organized structure, you're having a nucleus, then you have a complete distinguished feature you have, and when you are having a DNA, it's completely folded into what you call nucleosome, okay, what you call chromatin, right? The chromatin means the DNA and the protein together, you call it chromatin. Then that again, the DNA is wound, what you call in terms of the DNA is wrapped around the proteins called histones, and they have so many functions. Then those all serve in different mechanisms, including mitosis, meiosis, and so on. So as I was saying about the chromosome, of course you know about much of the chromosomes, like it has a well organized structure. What you are finding here is more of metaphase. You are generally finding in the metaphase, right? So in the metaphase you are finding, but generally the genome is organized. When you see the prophase, something like this, it's not organized much. And it has some irregular body, but when you see in the metaphase during the division, you are finding such kind of structures. So these are the different organization of the chromatin, okay, where you are using some packaging proteins like what's called, I was saying, histones. So histones are something like this, okay. There is something called bead, thread around the bead. When you are finding such kind of chromatin, such kind of long DNA, which is around 3.2 billion bases, you need some way that it should be properly folded. So there is something proteins called histone proteins. That's what I was saying. You see, when you are having a DNA, now the DNA has to be properly packaged. You cannot leave the DNA unorganized, where it helps in so many functions. So you cannot help make it as disorganized. So it is wound around particular proteins. What's called, these are called all the histone proteins, and all these are again arranged onto a what's called matrix. That's again, it's a protein. So all these constitutes, you see, all these constitutes. When you take DNA, then you have histones, then you have chromatin, what's called solenoid fibers, and so on. Then you are coming up with chromosome. That's what you are finding it a good organization. So you see the nucleosomes, when you are finding the DNA and protein, you have different types of histone protein like H2, H2B, H3 and H4 proteins, and so on, and around 40 base pairs of DNA is wound around the nucleosome. So this is how a nucleosome generally looks, where you have a linkers and so on. So this another histone. There are different types of histones. So this helps in packaging of the DNA. So this is what, and it also helps in the condensation, right? So I hope you understood a basic idea of how the genome is generally organized in prokaryotes and eukaryotes. Eukaryotes, you require really a proper higher level, complex level of packaging when you are comparing with the prokaryotes. Thank you so much.